Okay, thank you. We move on to our next talk, and the next talk will be by my co-chair, Giulia Gallo. Giulia works at the Perbard Institute, and her talk will be about zoonotic spillovers of coronavirus, specifically on virus entry. So, Giulia, please go ahead.
Okay, thank you. So let's move on. So alphacoronaviruses are endemic in human and animal population, and they cause different degrees of infection. See, here I'm showing you some representative, and as you can appreciate, the highest diversity of alphacoronavirus is found in bats. However, transmission and propagation of these viruses is poorly characterized, but we know that understanding the biology of infection is fundamental for preparedness and to counteract future epidemics. And when we are talking about the viral cycle, the first important step is viral entry, and for alphacoronaviruses, two different receptors have been described so far, APN and ACE2. So during my talk, I will answer three questions. So do bat alphacoronavirus use those same receptors? And how likely is their transmission to domesticated animals and humans? And what can we learn from human alphacoronaviruses that use those receptors, but we know are bat-derived? So for the sake of this talk, I selected some representative of alphacoronaviruses. Here you can see them. And to reflect, I tried to pick viruses from all over the world. And to reflect the diversity in bats, the majority of those are indeed from bats. And if you have a look at the identity matrix of their spike, which is the protein I've been talking about, you see it's quite low, around 60%, which is even lower when we look at S1, where the receptor binding domain is located. So how broad is APN usage among alphacoronaviruses? To answer this question, we took the spike genes from GenBank, we cloned them, we synthesized them, cloned them in a plasmid, and then expressed this along with structural protein of a lentivirus system in order to produce pseudoviruses. After 48 hours, we recovered the pseudoviruses and we used them. The first thing I've done was to purify these viruses to see if the spikes were properly expressed and could pseudotype. So here is our negative control. Our spikes are tagged, so we can see them. And P24, it's our internal control for purification. And as you can see, all of them are well expressed. We have an exception. This was not expressed. It might be to the poor quality of the annotated sequence in GenBank. And also I had some sequence from other animals, so ferret and shrew and rat. And so we can conclude that most of them pseudotype well. The second question, the most important one, was how broad is APN usage? So we took these particles and we used them for infection. And we infected cells that were transiently transfected to express APN that were derived from different animal species. And here I'm showing you our result. And as you can see, APN usage is not widely adopted by alphacoronaviruses. However, you can see here we had a positive virus, which is a bat, WA1087. So how broad is the receptor usage and the host range of this virus? We had a library of APN, of mammalian APN. Here I'm showing you flow data, so you can see that they all express quite well, and they are derived from 5 different orders of mammals. And here is our screen. So you can see it's APN of just 6 species is used, but we can clearly define if we look at the phylogenetic tree, we can clearly define that only the APN from ruminants and bats is being used and not those from primates or carnivores. What's even interesting is that the highest values we could get was with the APN of black flying fox. And this animal is geographically located in the same region where the sequence of this virus was isolated and identified. Then I told you earlier that alphacoronaviruses can use different receptors. So in order to better understand its potential for epidemics in humans, we wonder if this virus could maybe use another receptor to infect human. So we took human cells and we tried to infect them with our pseudovirus. And we took 229E, which is a human alphacoronavirus as a positive control. And you can see 229E infects well, mostly human cells, but all cells were negative for the infection with WA1087, suggesting that there is no infection. So what are the molecular characteristics of the interaction of WA1087 spikes and RBD with APN? We have a crystal structure of 229E RBD interacting with human APN. And what's interesting about alphacoronaviruses is that they can use different interfaces to interact with APN. Here I'm showing you a porcine alphacoronavirus. So thanks to AlphaFold, we could predict the RBD architecture of WA1087, and I superposed it with the known structure of 229E and PRCV. So here you can see that it's superposed quite well, but here it's where IPN is located. So this is an unlikely region of interaction because this loop will be embedded in the IPN. Sorry. Okay. So then I did the same with the porcine RBD. And although it superposes quite well here, the loops are totally all over the place. What's good is that recently AlphaFold got more powerful, and I could submit the RBD of WA1087 with the full APN, and I submitted the black flying fox. And as you can see, the prediction, it's different from what we know here. We see that the RBD is flattened on the APN. So just to remind you, this is a prediction. I'm not saying this is what's happening, but this gives us an idea that maybe the site where WA interacts superposes, or the interface is similar to 229Es. We also tried to identify key residues that could define or strange, but for now, I couldn't find anything really clear. I told you alphacoronaviruses use APN, but what about ACE2? So we took again our library and we tested against a library of ACE2 from different animals that we had in the lab, but everything was negative. But we were still interested in comparing the usage of IPN and ACE2 in the alphacoronavirus genus. So to do so, we focus on human alphacoronaviruses, 229E and L63, because they use APN and ACE2 respectively. So a bit of history. There is a 229E-like virus that's endemic in bat population, and it is believed that it jumped over to intermediate host, where it probably lost the NTD of the spike, and then eventually jumped to human, and through a combination got adapted, and that's the virus we are infected nowadays. As I told you, we know the interface of interaction of 229E RBD with human APN, and now we use the pseudovirus of 229E against our library of APN, you can see that the host range is quite restricted to just 4 mammalian species among those that we tested. For NS63, the history and the story is similar. So we have NS63-like viruses in the bat population, and it is believed that those encounter 229E-like viruses, and through a combination, a new virus came which had the genome of NS63, but the spike gene of 229E, then probably went to intermediate host, to borrow a combination, it finally mutated to jump into human, where now it's adapted and it's endemic. So the interface of NS63 RBD with human ACE2 is known, and in the lab we had a library of ACE2, and here I'm showing you what it looks like. So you can appreciate that it's completely different from what we've seen with 229E. And actually, it's much more similar to other coronaviruses that we now use ACE2. What's also interesting is that we see similar restriction as for other coronaviruses. So in primates, we see restriction with New World monkeys. And for carnivores, we see restriction with the Mustelidae family. So what are the molecular features that could define the host range of these two viruses? We know that coronaviruses RBD have 3 loops of interaction. And this is what you can see here for 229E. And let us focus on this loop, the second one. So it is known that there is a sequence region that has been described to account for host range. However, due to our data, we know that this only partially is true. And what's also interesting is that there is an extra motif in some of bats' APN, which could maybe account for host range. We tried to determine host range based on key residue, but we couldn't define it. So our hypothesis is that key residue may depend on the animal order. If now we move to NL63, again, we have 3 loops of interaction. And here, my job was much easier because I could clearly see which residue may account for host range. And what's even more interesting is that if now I superpose the RBD of NL63 with SARS-2, you can clearly see that loop 2 of these RBDs align quite well. And the key residue that I'm showing here, it's also known to determine host range for SARS-2. So to conclude, we show that some bats acquire the usage of APN, but not ACE2. Through molecular prediction, we could narrow down what might be the site of interaction, and it probably overlaps the one of 229E. And human alphacoronavirus display different host range depending on the receptor they use. And what we want to do after is to validate this key residue at the interface of interaction, characterize the dynamic of interaction, meaning we want to define what the affinity is for those interactions. And finally, compare data to other coronavirus genera, and all in all, to define what are the features that could predict potential spillover of coronaviruses and which might be the species that are susceptible to that infection. And so with that, I want to thank our group. And I just want to flag that other people from our group are going to talk. Naz is going to talk now about sarbecoviruses. Joan is going to talk tomorrow about sarbecoviruses as well. And Agnia has a poster on another aspect of the same project. And that's all. Thank you.
Thank you, Giulia. That was very nice. The paper is open for questions. Yeah, I see a question there. Yeah.
Thanks very much, Giulia. A lot of work. I'm just curious so 229E does use APN, we know that, but by presuming that bat-borne viruses that are 229-like may also use APN, are you going to lose out on potential novel receptors that these bats might have?
So this is just a subset of sequences I'm showing you. There are 30 more that we have been analyzing and do the same work. And in our bank, we have a 229E-like virus that I know uses APN. So what you're saying is true. I understand what you're saying. So what we want to do is all these viruses that you've seen here and more, we have been testing for the infection of human cells in case there will be a new unknown receptor that this virus may use. And what we want to do now, we have a collaboration, and we would like to get some bat cells to basically do the same. We also have a collaboration with a screening platform that could help us identify new receptors, human one. Unfortunately, there is no such tool for bats, but just infecting bat cells will give us an idea of how broad, like in a more, this is really in vitro because it is a pseudovirus, and human cells we are using. Using bat cells will make it more natural, like. That's great. Thank you.